Let me move this. Okay, so my name is Will. I'm a postdoc at Berkeley, and I will talk today about PONT, a protocol for online neuropsychological testing. A little bit of introduction. The literature is focused mainly on the cortex when we look at different domains of studies, from attention to arithmetic to motor sequence learning, while the contribution of subcortical brain mechanisms to cognition remain unclear. Parvizzi from Stanford called it the corticocentric neuroscience bias. So subcortical structures such as the basal ganglia, the cerebellum, the thalamus, and more might also be involved in cognition. And one solution to this problem is to do more studies that involve patients with subcortical pathology. So currently I'm focusing on two subcortical regions. One is the cerebellum, using patients with spinocerebellar ataxia, SCA, and the other subcortical region is the basal ganglia, BG, using patients with Parkinson's disease. But we have another problem with this solution. We are dealing with a very limited resource. We are dealing with the patients themselves. First, it's very difficult to identify and recruit these individuals to our project. But let's say you succeeded to recruit them to your experiment, their ability to participate in a research project is very restricted by their medical conditions, for example, their motor impairments. And as a result, if you will do a literature search, you will find papers with very small sample sizes, such as 6, 7, 8 participants. But not only that, it can take quite a bit of time to complete a single study, let alone a package of studies that might allow a more comprehensive story with appropriate control participants. And just as an indication to show you how much the problem is serious, if you will do a PubMed search results and you will enter two key terms in PubMed, one is neuropsychological studies, the other is fMRI studies, this is what you will get. So y-axis is the count, the number of results, the x-axis the number and the year. You can see how many more results we get for fMRI compared to neuropsychological study. And I think the trend is also very clear. So to address these problems in my first year in my postdoc, I've developed this protocol, a protocol for online neuropsychological testing. I like to call it PONT. PONT is a bridge in French, by the way. And as a first step, I recruited a team of research assistants that helped me to build this protocol. And the PONT protocol involves 5 main steps. In step number 1, we reach out to support groups from all across the United States, from Hawaii to New York, and we wrote an email to the group managers asking to transfer the information to the group members about the project. And in step number 2, each one of the group members reached out to us saying that they want to participate in our project. In step number 3, each individual is doing an online neuropsychological assessment in which we assess medical conditions, we assess motor abilities, cognitive abilities. It's about 1 hour session. And in step number 4, each individual is getting an email with a specific link and a specific ID, and you can do the experiment whenever they want. When they complete the experiment, we ask for some feedback and we pay them for their participation. This is the 5 main steps of PONT. In the first 10 months of the project, we were able to recruit about 420 participants for a database, 150 Parkinson's disease, PD, 150 SCA, spinocerebellar ataxia, and 120 control participants. And the goal of the project was to do a different experiment to assess both cognitive abilities and motor abilities. And using PONT, the idea is to assess different neural mechanisms in a wide range of abilities. And now I'll focus on the motor project. So this is a concrete example of a DSP test, a discrete sequence production test, in which participants are asked to put their fingers on the keyboard such that each finger is on the appropriate button as following, and then they need to execute the sequence as fast as possible. A little bit of background about sequence learning. Most researchers focus on the effect of memory on sequence execution, basically by comparing between repeating sequences to random sequences. And there is a lot of studies, large literature on the involvement of basal ganglia, BG, and cerebellum on sequence execution. Though there is one caveat here, two studies by Rothenberg recently have suggested that the PD, the Parkinson's disease literature is equivocal, meaning people show mixed results. Sometimes people with Parkinson are impaired and sometimes not. And as I see it, there is 5 main factors involved in sequence execution. One is perception. You need to visual ability to have visual pattern recognition of the digits. Then you need to create SR mapping, stimulus response. So you need to create the form, the association between the digit and your motor response. Then you have memory abilities. You can retrieve the previous instances of the sequence. Then you have other more general algorithm abilities such as planning of the sequence. And in the end, you need to have motor execution, to execute the sequence. And we asked 2 questions, 2 main questions. One, is it possible to conduct relatively complex motor sequence learning tests online on patients with neurological disorders? It was not clear because it's the first project, as far as we know, that try to do this kind of complex tests online on patients. And second, we asked more theoretical question, what is the contribution of the basal ganglia and cerebellum to memory and algorithm processes involved in sequence execution? And the method is the following. We've used a discrete sequence production test, DSP test, in which participants need to execute 4 key buttons in a specific order using 2 hands, so 8 fingers. And we had 2 conditions. One is the repetition condition, in which participants saw 8 different sequences that repeat 24 times. So in this condition, we assess memory-based learning. And the second condition is the no repetition condition, in which participants saw 192 different sequences. So in this condition, we assess more general algorithm-based learning abilities. And the main dependent variable is the execution time, or what people call ET. And this is the time from the first key press to the release of the last key press. This is the slide from the real test, so you can see the instruction. This is the way it looks for the participants. So they see the sequence, they need to execute, and they got feedback if it's correct or incorrect execution of the sequence. And in about 1 month, from the moment we started this experiment, we were able to collect data from 17 individuals with spinocerebellar ataxia, SCA, 21 Parkinson's disease, and 20 control participants. And this is the data. The y-axis is the ET, execution time of the sequence, the x-axis, the cycle of learning. And I think you can see 4 main phenomena. One, you can see that both patients group, the Parkinson's disease and the SCA, in general are slower than the control participants. And this makes sense, right? Because they have motor impairments. Another phenomena, you can see a nice learning curve. So control participants are able to learn the sequences. Third phenomena, you can see more benefits from learning in the repetition blue condition compared to the no repetition green condition. And this makes sense because in the repetition, they have memory benefits. And you can also see already in this figure that the ataxia, the SCA group, have serious learning impairments. They are not really able to learn this test. I am mentioning the obvious here because I think when someone is doing a new approach on doing patient studies online, it's important to show a good sanity test so that data makes sense. And I think this is quite convincing. So afterwards, we wanted to assess the learning benefits. So we calculated for each participant the slope for each individual, for each condition. So y-axis is the slope, x-axis the groups. And you can see first that the SCA group in green has less learning benefits compared to the control in blue. And the Parkinson group in pink have less benefits only in the no repetition condition, selectively. We have also wanted to exclude potential confound. So you can see that there is no execution time, ET and RT, response time for the first key press trade-off, and there is no ET and accuracy trade-off. But I think a nice feature of this figure is to see how much data, patient data, we can collect using PONT. So just to summarize these results, the literature suggests strong evidence for impairments in the SCA group, and the PD, the literature show mixed results. And our added value, theoretically, is that indeed SCA shows the classic impairment, but also when we do the experiments online. And the PD impairment is selective and depends on the algorithm needed, maybe some kind of preparation processes. So I started with two main questions. One, is it possible to conduct complex motor sequence learning tests on patients online? And the answer is definitely yes. Second, I asked, what is the contribution of the basal ganglia, BG, and cerebellum to memory and algorithm processes that involved in sequence execution? And the answer is BG probably is involved in motor algorithm, maybe planning of sequences, and cerebellum is involved in motor memory and algorithm processes of sequence learning. But I would like to finish the presentation with the advantages of PONT, of this protocol. And I think the key term here is accessibility. Using PONT, we have much more access to patients' population. And I think it's super important in the long run because it will allow first bigger sample size on patient population. Bigger sample size allow better representation of these populations, which a lot of time are biased in the literature. Of course, it will allow also more studies, as in all online studies, which will allow better understanding of diseases such as Parkinson, but also better understanding of the brain, such as the cerebellum. And I think a nice feature, at least from the feedback that we are getting from the participants, that it's quite engaging. It's much more engaging for the participants from the in-person studies. And in general, they enjoy doing the studies. Thank you very much.
Awesome, Will. So we have a specific question for you. Camila was asking, how long did it take for you to recruit your sample? And along with that, maybe you can talk about how the methods you're using to retain kind of participation with these patients.
Sure. So Camila, first is in about, let's say, now we are 1.5 years after I started my postdoc, after I started this project, and now we have about 500 participants. After 10 months, we had 420 participants, so 150 Parkinson disease, 150 SCA, and 120 control. So this is about the timeline of how much time it takes to recruit the participants using this approach. JT, your second question is, what is the second question? How to maintain the participants in our database?
How do you retain kind of participation with these patients or maintain a relationship?
Yeah. So first, we are doing the online neuropsychological assessment. So I have a team of RA, of research assistants that are doing online video sessions with the participants. And I think the personal relations here are super important because once people feel more connected to the study, more connected to the lab, they have more motivation to participate and do the studies. This is one way. Another way, we are sending them a lot of time the results if they are interested, their individual results. And this way we are connecting them over time, right? There is a more continuous relationship between us and the participants. And I think in general, the patients are super motivated because they want to understand the disease, they want to learn about it. So they want to contribute to studies that helps to understand the underlying mechanisms of Parkinson or ataxia.
Great. And another question for you, Will. So there's some in-person like assessments that require people to like move so you can assess their movements and even like maybe their reflexes. How were you able to adapt these clinical tests online?
So this is a relatively big operation. It's a very complicated operation to take, for example, we have done the medical questionnaire, which is relatively simple, but we have also the MOCA test, which assesses general mild cognitive impairment, and we have also a motor assessment. So Parkinson's disease are doing UPDRS, and ataxia are doing a SARA test. So in this test, we needed to go item by item and see if we can do it online using the video camera. So one of the tests, for example, is the finger to nose test, in which participants need to put their finger and to touch their nose. So we needed to think how to do it. So this is, and in the end, for example, in the SARA, we have done actually all the items online. And the results are exactly the same as the results that people have done in person, the average at least.
Awesome.